Genetically engineering on genetically modified food. Why is DNA important in genetic engineering? DNA is considered as a recipe of life. DNA can be found in the nucleus. DNA has 4 subunits: adenine, thymine, guanine, and cytosine. These 4 subunits help spell out instructions on how to make proteins in the organism grow and live. Small segments of DNA are called genes, and they hold instructions on how to produce a single protein. DNA has become a universal language because the genetic code means the same thing in all other organisms. This is why it is important. DNA is very critical for genetic engineering because they explained it as, when a gene is taken from one organism and is inserted to another, it gives the recipient organism the ability to express the trait that was taken from the first organism. Why are proteins important in genetic engineering? Proteins do all the work in the cells. They are a part of numerous of things in the cell, for example, cell walls, organelles, and etc. Not only do proteins help regulate reactions that take place in the cells, but they also act as enzymes that can help speed up reactions. Everything within an organism is made of proteins or is a result of a protein reaction. What is the goal of genetic engineering? Genetic engineering has one goal and can work by physically removing a gene from one organism and inserting it to another. This helps reach its goal of having the last organism have the ability to express the trait that is encoded by that gene. Steps to genetic modification. Step 1. Find an organism that naturally contains the desired trait. For example, corn. Corn is one of the most known crops that are genetically engineered. If they lack in fiber, farmers would need to genetically modify the crops to have more fiber. Step 2. Extract the DNA from that organism. It's like taking out the entire cookbook. Scientists would extract the trait they want, which is fiber, from another source, which would be anything ranging from white beans to avocados. Step 3. One desired gene or recipe must be located and copied from thousands of genes that were extracted. This is also known as gene cloning. Step 4. The gene then might have to be modified slightly to work in a much more desirable way once it reaches inside the recipient organism. Step 5. The new gene or genes will be referred to as a transgene. This will be delivered into the cells of the recipient organism, which will then be called transformation. There are a variety of ways. Some of them are bacteria. The most common way to transfer foreign cells into animals or plants is by the use of bacteria. Bacterial carriers will make things easier because some of the scientists inject the bacterium agrobacterium to infect plants, helping it be a suitable carrier for delivering DNA to a new organism. Then it will put in a special solution to make the cell walls more porous. The gene that was picked will be inserted into an extra chromosomal DNA molecule, which is also known as the plasmid, and the new gene that goes into the cell walls will be transformed. Biolistic. This is a process by which the DNA that is picked will then be attached to a microscopic particle of gold, also known as the tungsten. The DNA-laden medical particles will be shot into the targeted cell by the use of pressurized gas. After this, they will use microscopic particles to help release the DNA inside the plant cells. Electroporation. This process goes by the target cells and it will then be engrossed in a special solution with the selected DNA. They will then create a short and intense electrical shock that will pass through a solution. This will create small pores or tears in the cell walls which can open the new genetic material and reach the nuclei. The damaged cells will then be placed into another solution that will help manage and repair the broken walls. This will lock the inserted DNA inside the cell. The DNA will then join as a plasmid and reach into the host and gradually make its way into joining the plant's DNA to help the host with a new gene. Other methods: gene splicing, gene silencing, viral carriers, and microinjection. Step 6. The transgenic organism is created. The traditional breeding will be used to help improve the characteristics of the final genetically modified product.